Good morning students. Today you are going to study about mitochondria. Before you are entering into the new topic, you can recall the definitions, plant anatomy, the study about internal structure of plants, physiology, the study about function of the plants. Let's see mitochondria. You know very well our body, plant body is made up of cells. The basic unit is cell. Cell consists of many organelles. One of the important organelle is mitochondria. Mitochondria is filamentous or granular. It is found in the cell. Scientist Kolliker in the year of 1857 discovered mitochondria. Mitochondria is also called powerhouse of the cell. The size varies from 0.5 micrometer to 2 micrometer. Then mitochondria contains 60 to 70% of protein, 25 to 30% of lipid, 5 to 7% of RNA and small amount of DNA and minerals. Let's see the important parts of the mitochondria. This is outer membrane, this is inner membrane, then finger-like projections are called cristae, then the matchstick is F1 particle, then the space matrix, the dotted one ribosome, this is RNA. Let's see one by one parts. Mitochondrial membrane. It is separated as outer membrane and inner membrane. 60 to 70 Armstrong thickness. Outer membrane, it is smooth and freely permeable. Freely permeable means it will allow all the substance to the matrix. It contains enzyme, protein and lipid. Then it contains a special protein, that name is called porine molecule, which acts like a channel for passage of the molecule into the matrix. Next one, inner membrane, it is semi-permeable. That means it will allow partly what it wants only it will allow. Then it will rich in enzyme and carrier protein. Then it consists of 80% of protein and lipid. Next, here cristae, the finger-like projections on the inner membrane of mitochondria, that is cristae, this one. So the finger-like projections from inner mitochondrial membrane, it will increase the inner surface area to hold variety of enzymes. So the finger-like projections, it hold variety of enzyme, that is cristae. Next here, the matchstick-like structure, that is F1 particle. Oxysomes, minute regularly spaced tennis racket shaped structure which involve in ATP synthesis. ATP is a energy currency. ATP, adenosine triphosphate. So the tennis racket shaped structure helping for ATP synthesis is oxysomes. Next one, mitochondrial matrix. This space, full space, that is mitochondrial matrix. It is a complex mixture of protein, lipids. It contains enzyme that needed for Krebs cycle, then ribosome, tRNA as well as mitochondrial DNA. So what are the parts it contains? Mitochondrial membrane, then cristae, oxysome, then matrix. Let's see the function. First point, it is a main organelle for cell respiration. It produces energy, more amount of ATP in the, that is the energy currency. So we can call this as a powerhouse of the cell. Then it is maintaining the normal concentration of calcium ion. Then it regulates the metabolic activity of the cell. Which one? Mitochondria. So mitochondria is the main organelle for cell respiration, maintaining the normal concentration of calcium ion, then regulate the metabolic activity of the cell. Next we can see the types of respiration. Types of respiration. External respiration, it's a physical process, just exchange of gases. Plants obtain oxygen from external environment and release carbon dioxide and water vapor. Next, cellular respiration. Here it's a biochemical process. Food is oxidized within the cell to get energy. Food is oxidized within the cell to get energy. This is cellular respiration. Next, aerobic and anaerobic. First, aerobic respiration. Here the organic food is completely oxidized with the help of oxygen into carbon dioxide, water and energy. Most plants and animals have this type of respiration. Here the food molecule with the help of oxygen converting into carbon dioxide, water and energy, that is ATP. Here aerobic respiration involves three stages: glycolysis, Krebs cycle, electron transport chain. Glycolysis is the first step for both aerobic and anaerobic respiration. Here it takes place in cytoplasm. Here 6-carbon compound glucose is converted into 3-carbon compound. The 3-carbon compound name is pyruvic acid. Here glycolysis is glucose splitting. Glucose splitting. Next one, Krebs cycle. It takes place in mitochondrial matrix. Here the pyruvic acid formed from glycolysis entering into the matrix and converting into carbon dioxide and water. It is happening in the form of a cycle. So we are calling this Krebs cycle as a tricarboxylic acid cycle. Next one, electron transport chain. It includes a system of electron carrier complex. It will be located on the inner mitochondrial membrane. Here two energy-rich molecules like NADH2, FADH2 formed from glycolysis and Krebs cycle or get oxidized and form NAD plus and FAD plus. While oxidizing time, it will release some energy as an electron. That electron's energy is used for synthesizing ATP from ADP. So the synthesis of ATP from ADP is called oxidative phosphorylation. So aerobic respiration involves three stages: glycolysis, Krebs cycle and electron transport chain. Next one, anaerobic respiration. Respiration without oxygen. Without oxygen. So glucose is converted into ethanol in some plants and lactic acid in some bacteria. So with the presence of oxygen, aerobic respiration. Without oxygen, anaerobic respiration. Next one, respiratory quotient or Q. It is ratio of volume of carbon dioxide liberated and volume of oxygen consumed during respiration. So ratio of volume of carbon dioxide liberated and volume of oxygen consumed during respiration, that is RQ. So now you know very well aerobic respiration, anaerobic respiration, external respiration, cellular respiration, and what is RQ. It is nothing other than respiratory quotient. Thank you.